Warm welcome to everybody. And I think we have participants from all over Canada and Sri Lanka, as well as from other countries, I think from Australia and UK. And very warm welcome to all of you for our fourth webinar. And I am Anura Herath, the secretary of the Alumni Association of the University of Peradeniya, the Ottawa chapter. And this is our fourth webinar of the series of webinars that we have been having over the last year and this year. And on behalf of all of us, I would like to welcome Dr. Dushanta Jayavira, who was very kind enough to accept our invitation for the second time, besides his very, very busy schedule. And very warm welcome to you. And he will be sharing his tremendous experience with us in a short while. Now, please let me remind you of some housekeeping rules for this webinar, as usual. And for your information, we are recording this full webinar. And we will be posting it on our website. So anybody who wants to watch it again, and you can do so. And therefore, during the presentation, we kindly request you, all of you participants, to mute the microphone and put the video off, and that will increase the quality of the presentation of Dr. Javira. And at the end of the presentation, there is a Q&A session where we would encourage all of you to post your questions on the chat line. So we will take one by one as we go along with the time permits, and we can put those questions to Dr. Javira, and I'm sure he will have very rich responses to those questions. Those are the main housekeeping rules. And then let me present you with the disclaimer of this webinar. I'm sure all of you can look at it. This webinar is designed to provide helpful advice and information on the subject of the novel coronavirus, also called COVID-19. Individual diagnosis and concerns should be directed to each participant's personal physician. The authors and the host are not responsible for any specific conditions that may require the medical treatment and specific supervision, and are not liable for any damage or negative consequences arising from this webinar. So this is a medical webinar, and I think this is important for us to have this disclaimer. So with that, let me invite our president of the association, Dr. Susanta Motalege, to give his opening remarks. Susanta, over to you. Thank you.
Good morning, good afternoon, and good evening to you all participating in this today from various parts of the globe. Today, we have participants joining us from all across Canada, the US, UK, Australia, and Saudi Arabia. Thank you all for being here today. It is my pleasure to welcome our speaker, Dr. Dushanta Jayavira, on behalf of the Alumni Association of University of Peradeniya, Ottawa chapter. We call it as AAU POC. Thank you, Dr. Jayavira, for accepting our invitation to present this webinar on the timely needed topic. First, I would like to debrief the objectives of our association. The main objectives of our association are to help students of the University of Peradeniya through scholarships. We also organize events and social activities to foster and strengthen our friendship among our members and the Sri Lankan community at large, both in Ottawa and beyond. Finally, we are helping our alma mater by supporting academic needs through special donations when needed. The AAU POC scholarship program is one of the main features of our association, which supports the undergraduate students at the University of Peradeniya by providing financial assistance. We usually offer 10 scholarships worth 360 Sri Lankan rupees per year, and we collect funds by organizing various social activities and by the generous donations from our members and well-wishers. Here, I would like to invite you to contribute to our worthy cause. If you would like to donate to our scholarship program, and if you are living in Canada, you can use our e-transfer method, or you can send a check payable to AAU POC. Once again, thank you, Dr. Jayavira, for taking your time to speak to us today. I would also like to take this opportunity to thank the organizing committee of this webinar, our moderators of this event, the technical support team, and the current executives for their hard work. Thank you, Dr. Nimal De Silva, our past president of AAU POC and his executive committee for the initiation of AAU POC's webinar series. Also like to thank all the past presidents, previous executive committees of the AAU POC, and our members for their continuous support. Without their help, we would not be able to carry out our duties for the association as such. Last but not least, I'm grateful to our webinar sponsors, Murugesh Gandhi from Mega Financing and Arantika D. Lambert from TD Canada for their generous contributions. We really appreciate your financial support. Your donations have greatly contributed to the AAU POC's scholarship fund. And now, without further ado, I would like to invite Dr. Harini Silva to introduce our special speaker to the audience. Thank you, everyone.
Thank you, Dr. Mohotalage. It is my great pleasure today to introduce our guest speaker, Dr. Dushanta Jayavira. Dr. Jayavira is both a clinician and is a researcher. So he has worked in the field of infectious disease for more than 20 years. He graduated from Faculty of Medicine, University of Colombo, and currently he's a professor in clinical medicine at University of Miami, and he oversees the infectious disease clinics at University of Miami Miller School of Medicine. When you talk about his research aspect of his career, he has been a principal investigator for both NIH-sponsored and more than 80 industry-sponsored clinical trials related to infectious disease. So when the pandemic started, he participated as an investigator in multiple therapeutic investigational trials, to name a few: a transfusion of convalescent plasma to limit COVID complications, the and he is also the principal investigator for the Janssen COVID-19 vaccination, or the Johnson & Johnson vaccination. We are very grateful for Dr. Jayavira to accepting this kind invitation to talk on this very timely and popular topic the current status of COVID-19 vaccinations, the facts and the myths. Please welcome Dr. Dushanta Jaivi.
They have been. Well, I first got to know them through the Sri Lanka Foundation, through Anura, and then now I've met all of you, and I'm delighted to be partnering with you in this webinar. As Shami said, that I'm a simple clinician, but I do research, and I've been doing it for some time. So today my mission is to talk about COVID-19 vaccines and give a little bit of background, talk a little bit of Sri Lanka. I mean, as you all know, we are struggling in Sri Lanka now with the COVID pandemic. It's very strange how things changed over the last 4 or 5 months. For the initial period when the pandemic began, Sri Lanka was so ahead of everybody else. In fact, my first lecture that I gave to Sri Lanka Foundation on COVID, I was praising Sri Lanka for the amazing work that they have done through the government and the NGOs and the physicians. And in fact, we wrote a paper comparing, myself and Saroj Jayasinghe got published a paper on Public Health Sciences book, one of the journals, comparing Vietnam, Sri Lanka, and their success. And something went wrong. And then it is what it is. And now our job is to somehow help Sri Lanka to get back on track. And I always, when I start a lecture, I say that compared to, no, I'm not talking about Canada, but compared to United States, I spent roughly $12, less than $12 for my entire medical education. And we just have to remember that that was all paid by poor Sri Lankans living there who are suffering now. So I just want to mention that before I start. So in this conversation, I'm going to talk about the COVID pandemic in US and Canada. I should have put Canada there also. And we'll talk about the vaccines, Pfizer, Moderna, then the adenovirus vaccine data, including the Oxford AstraZeneca, which is now called something else when India makes it. I have a slide on that. The Sputnik vaccine, the vaccine hesitancy. We talk about variants and summarizing. We try to do it within 45 minutes or maximum 50. I'm very grateful to your group for forwarding some of the questions ahead of time, so that I can give appropriate response, and some of the things I embedded in my slides as I go on, so that I answer your question. It's very important when we have a webinar like this that I'm here to respond to your needs, and you're not there to respond to my needs. So I'm here to share whatever I know with you, and if I don't know, I will get back with you and answer the questions later. So basically, as you all know, the COVID, we didn't know very much about COVID, except one of our Sri Lankan compatriots, Dr. Piris, who was in Hong Kong, Malik Piris, who had worked on this since leaving Peradeniya. Mind you, he's a Peradeniya graduate. And he discovered part of the coronavirus one decade ago. So the virus, this is an RNA virus. It enters through the angiotensin 2 receptor. Now, viruses are like some of the politicians in the United States. They have different plans. They have different doors to enter and take control of things. And like politicians, the viruses destroy the cell, and that's what some of the in US does. So the virus enters through angiotensin 2 receptor, but there are other receptors also there, which I'm not going to go into detail, that they could potentially enter a cell. Once it enters, what it does is that it releases the RNA. I'm sure that we did it in chemistry and biochemistry in the university, and the RNA gets transcribed and gets replicated, and this whole life cycle happens in the cytoplasm. Although I have a slide later which shows that with the reverse transcription, some of these things can get into our host genome, like in HIV, but it's not that required. It's optional, and then the viral particles get released, and then of course downstream all the signaling pathways cause ultimate chaos in through the cytokine storms. But basically this is what happens. So the vaccines are essentially trying to mimic this infection, so that you fool the system to say that, well, there is infection, but actually there's no infection, and the immune system starts responding, and then we have a immune response. So with RNA vaccine, so if you take a human cell, when the DNA polymerase makes a DNA copy of it, it has a proofreading mechanism which reads the copy that is made and see whether there are any mistakes and fixes it. So for example, when I'm basically an HIV researcher before COVID came. So one of the reasons why people used to call HIV is a really smart virus, because it was mutating like crazy, simply because of that. The reverse transcription that causes a DNA copy of the RNA, which is slightly different from COVID-19, doesn't have a proofreader. So every time the virus replicates, it keeps making mistakes. So every mistake is a mutant. So most of these mutants are suicidal mutations, where it makes a mutation and the virus cannot survive and dies off. But there are some mutations which are beneficial to the virus. And some mutations leads to like a increased replication capacity, some leads to decreased replication capacity. In general, when there is a survival advantage, then usually the replication capacity goes down. But so that was in HIV. So when we look at COVID-19, we were kind of expecting the virus to have mutations because it's RNA virus. And therefore we were expecting to have variants. So when people say, oh my God, there are variants, of course we were expecting variants. When vaccines were developed, people knew that things were different. So when they developed vaccines, they used the conserved part of the virus, so that even if there are variants, that the antibodies will be made against the conserved parts. And I said the viruses are like politicians, they know how to change, and they will make sure that somehow then escape some of these antibody activities, and some of them survive. Actually, basically, it's like, survival of the fittest. So there was a question by one of your colleagues from the audience. Please explain the airborne nature of the virus. So this is actually a cartoon which just shows that the virus could enter from anywhere, from eyes, nose, mouth. That's why we were told, or we were telling people, that wash your hands, be careful. These are simple things we tell our children from the day they were born, but we don't do it. But now we have to do it. So depending on the particle size, it can be aerosolized or not aerosolized. So if it's less than 5 microns, then of course it gets suspended in the air and it travels everywhere. On the other hand, if it is greater than 5 microns, then either it will fall on your feet or on your table if you cough, and then you touch your nose, touch your mouth, or it could go through your respiratory tract and enters into your lungs. So the question is, that is why that the airborne nature of the virus. Now, so you can ask, at the beginning, CDC said there is no airborne virus. There's no airborne spread. If you wear a mask, everything is okay. And if in the same room, if it's more than 6 feet, that there is no transmission. So here comes the variants. When the variants come, and if the infectivity goes up and the survival gets better for the variants, now the things change. Before I get to the next slide, I want to talk to you briefly about the stool. So there are angiotensin 2 receptor right down your GI tract. So what happens is that the virus can replicate in the GI tract. In fact, checking the virus in the stool is another way of diagnosing COVID-19, because it's sometimes people have most of the symptoms in the GI tract. And if you do a swab in the mouth, may be negative, but if you do the stool, it could be positive. So you have to be careful, as we understand more and more about the disease, that these things could happen. So in addition to that, Another epidemiological interesting tidbit is that so all these the stool and things like that goes into drainage, and there is pollution. So to find out the baseline incident prevalence of the disease, in fact, University of Miami is doing a study looking at the wastewater and looking at the COVID-19 in the wastewater. And then you can, depending on the zip code, you can say that this is happening here or this is happening there. And sometimes it may be you can do some predictive modeling to say that where the next, rising cases would come. So, then I go to the next slide. So this is a very interesting slide. This came from, just look at the date. This was January 2020. At that time, there was no transmission like that. So the Chinese reported saying that, so if you take these tables A, B, C, and D, and the people did not get infected across the room. So the authors noted that the results do not show that there's no long-range aerosol transmission in the space. But this is what January 2020. The world has changed. So that's why I say that some of the things we say are not correct. In fact, I always teach my medical students, 50% of what I say today will be wrong tomorrow. The problem is that I don't know which 50%. So you have to have a critical mind and always question everything people say, even your teachers, and do independent research and find out what exactly the truth is. So this slide comes from Sri Lanka. And then if you go to this website, which I'm going to click in a second, you can see the pandemic problem. This was on May 15th, 2021. In Sri Lanka, there were 140,471 cases. Now, cumulative cases per million population, this is again coming from similar website from the epidemiology unit. There were 6,900 cases, roughly about 48,000 deaths were about 48,000 per million cases. Per million population, compared to India, we are doing much better. But compared to, say, Australia, we don't do well. And the age distribution, you can see that, age distribution is quite greater than 90, there was a significant amount, 76 to 90. You can see that the younger the people, lesser the death rate. So if I click on this now, I'm going to show you a link to the other website. So you can see this is real world. So now you can see this is done by the Sri Lankan Health Promotion Bureau. Right now, today, there are 158,000. So it has gone up almost 18,000 over the last 2 weeks. You can see the devastation that is going. Today, there are 2,145 new cases in Sri Lanka, a daily rate. And also, if you scroll down, you can see the daily new cases, daily deaths. Thank God the deaths are less. We have done, so hats off to the physicians in Sri Lanka and the health system that prevents deaths. And you can see the case fatality rate. Sri Lanka has the lowest fatality rate comparison to all these rich countries. And of course, you can see the breakdown. So now I'll go back to my slides. Okay, okay. There you go. Okay, so I want to thank Manoj for teaching me this 10 minutes ago. Without Manoj, I wouldn't be able to do this thing. Okay, so now you see from 140 it went to 158,000 just within the last couple of days. So we are in big trouble. So this is a slide I made a couple of days ago. You already seen that. So this is the distribution of different places. So Colombo obviously have highest number of cases, 48,000, with Gampa having 28,000, and you can see that. So some of these things are based on population density, because, for example, if you take Mullaitivu, Kilinochchi, Mana, the number of people living there are relatively less. So it's less, as there are big cities like, say, Badulla or Jaffna or Kalmone. I mean, things getting little higher as more people live. So you can see the gravity of the problem that is happening. So I want to give another share now. Sri Lanka Foundation. So this is again this is the Sri Lankan Foundation who is doing all this work for different hospitals. For example, the donation requests that are coming from Gampaha and different places. So I just want to show you that. Okay, so I'm going to get back to my talk now. So so you get the picture now. You know that which is right. Now let's ask about the vaccinations. See, I live in United States where like 40% of the people hands down says I don't want to be vaccinated. I don't want to be vaccinated. So whereas Sri Lanka, going historically, looking at the vaccine history of vaccinations, have been excellent in vaccine acceptance. That is partly because we have a socialized health systems and we have a high literacy rate among the population. Thank God for free education and free healthcare. And the people are really understand the value of vaccine, and we have a mechanism to track it, and we have done it. In fact, this was too good to be true. In fact, I called Sri Lanka to ask, is this really 99%? I mean, this is too good to be true. But apparently this is what they are in the books, and that's what they report to UNICEF. So it is what it is. Even if you reduce another 2 or 3%, still we are ahead of the curve. So these are the vaccines that are available in Sri Lanka. Covishield vaccine. Covishield vaccine is like, a generic AstraZeneca made by the Serum Institute in India, in Pune, I think. So then you have the Sinopharm vaccine. Sinopharm vaccine is an inactivated virus. I tried to find out much about this. The problem is that they haven't published any phase 3 data. They have some phase 2 data, which tells you the immune response. But this phase 1-2 data is not very robust. We have seen over and over and over again that these in vitro animal models, phase 1, phase 2 data does not really pan out when you run a clinical trial. So you can't hang your hat on that, and you can't depend on that. So I'm not holding my breath on Sinopharm, but the reality is that it is better than nothing. It is better than nothing. I have some slides later on vaccine efficacy and durability with the influenza vaccine, and I get to that, I can explain why I think this is okay. And the Sputnik, we have that 15,000 doses, but compared to 21 million people in Sri Lanka, this is really sad situation that we can't get vaccines. We are hoping that we could get some Janssen vaccine to Sri Lanka soon. So let's get to the vaccine development, and I'll start with the mRNA vaccines. So this is from New York Times, the vaccine tables that pathways and remaining questions. This is what New York Times published. I mean, that's one place that they track things. So there were 4 abandoned vaccines, and they approved like 8 different people, not by the United States. United States has only 3, but you can see the things on the left-hand side of your screen. The Pfizer BioNTech is excellent. Then the Moderna. Then there's the Russian vaccine Gemalaya, which is also called the Sputnik. AstraZeneca is not approved by United States. J&J is approved. And so you can see that there are multiple other things being worked out. And I have some interesting conversation that I'm going to have with you later on about how to actually know which is which. So I think I prepared that because one of you asked that question, how do you compare all this? And that's an excellent question, and I have an answer to that later on. So what's the time duration of antibody response to antigen? So generally, if you get exposed to antigen within like 14 days, you get a primary immune response. So it's interesting because they have tracked down some of our vaccines, that when you give a vaccine, that you can see the primary antibody response within 14 days, and then when you expose again, then the second response is much more robust with higher antibody response, generally about 4 to 6 weeks. So this gives a rough estimate as how long it takes for a good response. So and then the question, one of you asked, how long does the COVID vaccine stay effective? So we don't know about COVID vaccine. The data has been tracked in Moderna and Pfizer. They started their vaccine in July last year, end of July last year. So they have gone up to about 8-10 months, and they have a decent antibody response. They have not published it, like everything else during Corona time. Things are just leaked out to the press, partly to improve their stock price, but partly to release results. But I don't think it's a good thing to release results like that, because it gives you the wrong impression. And when the real data comes, you realize that, my God, what they said was not true. I guess, in the stock price world, that they don't care about the truth. They care about how much money they make. So if you look at the left side of the graph, there is the days of between vaccine and vaccination and onset and the vaccine efficacy. You can see that there's a large confidence interval. So you can be here or here, and then this is the median. And you can see that initially you can get about 40% protective effect, but towards latter, it's about 20-25%. So when people look at Janssen vaccine and say, oh my God, Janssen vaccine is not as good. If you took the flu vaccine, Janssen vaccine is like almost like 3 times better or twice better than flu vaccine for the same kind of effectiveness. And then if you compare the duration or the durability of activity, predicted risk, then you can see that the durability is about 150 to 180 days, which is roughly about 6 months. So flu vaccine lasts for about 6 months. That's one of the reasons that when there's a kind of a debate about, when you get the flu vaccine, if you get it very early in September, then by the time about March next year, especially in Canada where the winters are long, whether you need another booster injection, especially if you're elderly, where as we get older, our innate immunity that generates antibodies become weaker and weaker and weaker. So that's why as we get older, everything starts break down. Even a Lamborghini breaks down after a couple of years. So that's what happens to us. So I'm not saying a Lamborghini, I'm more like a Fiat, but so the immune response in the older people are less, and the duration of the vaccines are short. So that's why in flu vaccine there are some controversy about should we give a second dose. Definitely, as elderly, we are given a high dose vaccine compared to low dose vaccine. So there was another question: what is the time duration between the 2 vaccines by one of your colleagues? Basically, I put the 4 common vaccines: the Pfizer, Moderna, Janssen, and Sputnik. And on the right hand side, you can see the interval is roughly 21 days to 28 days, whereas the Janssen is single shot, so there's nothing to worry about. In fact, there was a very interesting article from AstraZeneca, which I should have put, is about, again, 3 weeks, that if the AstraZeneca vaccine was delayed in older people, they had a better response. So they're trying to figure out, how this happened. So, but you have to be careful. This subgroup analysis of data sets are very dangerous, because sometimes you get the wrong information, the wrong interpretation, if you really don't know how the study was done. So it's very important that read the fine print, so that understand what the methodology is. So so here is the meat of the talk. How effective is Pfizer? Pfizer is 95% effective. It starts off 7 days after second dose, so it's pretty good. P-value is like through the roof. and mRNA vaccine with Moderna is 94%, it's no difference. And so it really, there's nothing to, this is really a home run, or in cricket sixer, right? There's nothing to argue with this vaccine. So there have been a question like, what if you had coronavirus infection before, and would that make a difference if you take a vaccine? Should I take a vaccine if I had corona before? So the general principle is that it is better to get the vaccine because we don't know the decay of the antibodies after infection is pretty rapid. For example, Ebola, the antibody decays have been dramatic once they recover the Ebola antibody levels. There are multiple publications from West Africa, how quickly it drops down. Not that the vaccine antibody response are remarkably better, but they're better. So you would say that, okay, even if you had the infection, wait for about 2-3 weeks and get the vaccine. The second thing is that will it be good or bad? There's no difference. So this study showed there's no difference between if you had an infection before or didn't have an infection before. If you get the vaccine, you get the similar response. The question would be, would it be more durable? That's the interesting question. We don't know. So there are research going on. In fact, we are doing a study about looking at that in Miami with one of my colleagues, Savita Pava, trying to see whether this could be tracked and meaningful information can be obtained. So the other one was like, what are the side effects? 80% of the people get pain in the injection site, redness, and then if you're doesn't matter whether you're young or old, you're same thing happens, swelling, and it could be pretty severe also, but most of the time it's mild. So it's nothing really. I'm sure most of you have had the vaccine, and I have had it, my kids have had it, my wife has it. They're pretty straightforward. And then this is again looking at the mRNA vaccines, which includes the Moderna. This is from Moderna. Again, similar system, we get myalgia, nausea, erythema, induration, pain, headaches, chills. These are things that you get in a head of a mild attack of flu. They're actually much better than the mild attack of flu because this lasts only like 1 or 2 days. You take 2 Tylenol and it's go away. In fact, the AstraZeneca was having this thing quite severe. So the Oxford people changed the protocol to add 2 Tylenols the next day to mitigate the symptoms. It's kind of strange because this is an adenovirus vector, and Janssen has adenovirus vector. We didn't see that much side effects as much as in AstraZeneca. Maybe people were expecting it. So the emergency authorization of Pfizer vaccine was given, and I put this slide, not that you haven't heard about it, but I want to talk about the polyethylene glycol, which is thing that's stabilizing agent and preservative. And so you have to actually look at this, because if some people have had allergy to some of these things, you can have allergic reaction. So you have to look at the other components of the vaccine to stabilize and create the durability. And some of these things could be potentially the cause of the vaccine, the allergic reaction, not the vaccine itself. So why is it important? Because when you have the next vaccine, if it has a different formulation, maybe that you can take it without kind of mislabeling yourself that you are allergic to vaccine. So it's very important. You have to characterize your reaction exactly what it is. So again, the Pfizer went ahead and did a study on the 12 to 18 and didn't have any side effects, and it was approved. So I think this is pretty good. I think it's been approved for people who was age of 16. So Pfizer vaccine has been approved. So the question is, can you take Moderna? Maybe, but if you just stick to data, which I strongly recommend, because these days you can't listen to news or anything on the internet. It's all garbage. And in fact, the comedians give the news, and the news gives jokes. So, I mean, you don't know who to believe. So, okay, so let's get down to adenovirus vaccine data. So, so the most viral vaccines, like vector-based vaccines, are either attenuated or inactivated viruses. So they are, if you're using that route, because it's easy and inexpensive. That's why, like, Sinovacs and some of these vaccines are attenuated or inactivated viruses. So what happens is once the adenovirus is inside, the viral vector comes inside the cell, it hacks into the molecular system, like any hacker that gets into your system in the computer, and it pretends that it's a natural infection, provokes an innate immune response. I had already told about the innate immune response, triggering inflammation and mustering like a B and the T cells. B cells give to humoral antibody, T cells give to cellular antibody. That means lymphocytes and things like that on the T cell side. Downside of this thing is that if you already have antibodies to cold virus, when the vaccine hits, the antibody can take the vaccine off. So I think this was carefully studied by all the people who are using adenovirus. So what they do is that they find the strain that the normal people don't have antibodies. For example, AD26, which Janssen uses, or 85 that the Russians use. Or Russians use 85 and 26 together, or the AstraZeneca uses chimpanzee adenovirus. So they do seroprevalence study across the nations, across the world, and they map out the entire world and say, okay, these areas have these antibodies, these areas have these antibodies. So you can't use these adenovirus vector vaccines if there is underlying antibodies against adenovirus. So for example, sub-Saharan Africa has a significant amount of antibody against adenovirus. So we wouldn't advocate putting any of these adenovirus vaccines on sub-Saharan Africa. But the population in sub-Saharan Africa is not that great. I mean, I'm sure that we can manage that with Moderna and Pfizer. So that's something that's important. So in the United States, Africa, Canada, Europe, Australia, the antibodies against adenovirus AD26 is very low. There are some reports that in South America that there may be a little more, but that has to be determined. So when you use a vector to generate a vaccine, this is like Nikolai Petrovsky from Flanders. This is from the scientist's website. It's a very reputable website. With vectors, you are always trying to find sweet spot. If it's too weak, they don't work. If you're too strong, they are too toxic. So this is where the phase one, the animal experiments, preclinical data tells you that what works and what doesn't. I mean, you can try it on a test tube also, but or tissue cultures, but basically you're looking at the animal data and then the phase one, phase two. So Janssen, as I mentioned before, is AD26 and AD26 neutralizing antibodies are uncommon in Europe and US. About 10 to 20% having antibodies. That again, it's not a significant antibody response, so we are not worried, whereas sub-Saharan Africa is about 80 to 90%. Sputnik, I mean, we although we are very suspicious about the Russians that they don't share much data, but their method was actually pretty cool. They would start with that AD26 vaccine virus vector, and then you switch to 85. So I think it's a pretty cool method. And let's see what it shows with the real world. I mean, I'll show the data later on the Lancet paper. And then again, as I mentioned before, chimpanzee adenovirus is used by the AstraZeneca and Oxford, which is again a pretty good system. So this is a study that Janssen study, the ensemble study, which I was the PI for University of Miami. It's a randomized, double-blind, placebo-controlled people who are 18 years. We enrolled 44,000 in Miami. We put about 400 because we didn't have time and because it was so fast. It was recruited so fast. And the efficacy rate was at day 28 post-vaccination, 66% overall across the world had efficacy versus 72% in the United States, because we much better. Obviously, we had less adenovirus antibodies, and also we have better healthcare system. And in a study, I'm not saying that the United States have better healthcare than other countries. I'm saying in a study, we did better. That's completely different. And 57% in South Africa with the variants that didn't go down very well. But I'll talk about it later because there are different ways of looking at this, and you will see that Janssen doesn't look that bad when you look at the data in a more critical way, which we will discuss a little later. So if you look at preventing severe or critical disease, 85%, and prevent death, 100%. So basically, what I'm really interested in preventing myself getting admitted to hospital, and so almost 85%, and if I'm going to prevent from death, then it works. I mean, it's funny because yesterday I had a patient in my clinic, and I told him that, ask him, he's African American. So among African American, there's a tendency to distrust the health system because of the social disparities and because of the terrible social determinants of health in the United States compared to Canada. You guys are in a much better place than us. And Sri Lanka. In Sri Lanka, social determinants are much better. So the patient says, Doc, I don't want to because I know so many people who got sick when they took the vaccine. So my response was that, oh, that's true. Yeah, I know. But I said, for everyone you got sick with the vaccine, I can give you 100 who died in our hospital. And I started explaining this results. Within 2 minutes, he said, okay, give me the vaccine. Right. So it's very, very simple that, when people who anti-vaxxers, especially a certain party in the United States who doesn't believe in vaccine, doesn't believe in anything, talks like that. And when you talk about facts, then they realize that facts show something else. So here's the Janssen vaccine data. So looking at the moderate to severe to critical cases between the placebo and the vaccine, you can see that there's a tremendous difference in the response. I'm not going to spend too much time because I have 3 slides like this. The next one was severe to critical compared again. There's a huge gap, so this is much greater. And then they're looking at South Africa. Still there is a significant difference. So I'm not going to go in detail. These are the survival curves. So obviously the placebo has less survival than and adenovirus. And actually, sorry, it's not the survival, it's the cumulative incidence of cases. Sorry, cumulative incidence. Incidence is much more than placebo than the adeno and this thing. Sorry about that mistake. So then if you look at the side effects, I'm not trying to say that this is better than mRNA vaccines. I think you must never compare one study against the other. That is fundamental no. If there are any students listening to this from undergrad or medical school, you never ever compare one study to the other because the populations are different, the statistical methods are different, how you define the side effects are different. So you have to say, in this study, this is what we found. In that study, that's what we found. Definitely these are apples and oranges. So, but they are both fruits. So you can say that, they are both tasty, I would say. So now we got a little scare with Janssen vaccine about this platelet thing. This timeline because I'm in the study. So we had a meeting with the FDA before it was lifted, and this is what's shown by the FDA. And so the emergency authorization of Janssen vaccine was given in 27. They were recommended by 58. Vaccination starts on March 2nd, and then 6 cases. This is a little old slide now. There are about 8 cases, and then the FDA puts a pause. Now, so this demonstrates 2 things to a general audience. First is that FDA is pretty good at tracking this. So I'm not just kudos to FDA. I'm not saying that FDA is very good on everything they do, but they are good. And not only FDA, all the the European regulatory agencies, they all are very good at tracking. The question is that were they right to stop the study? I'm not sure. because it was about 7 to 8 close to a million, 1 to million ratio. I don't think it's a huge deal, but I think there was I understand why FDA put a hold, because unlike other vaccines, these were rushed through 1 year. So, there were a lot of deliberations and discussions. Are we doing the right thing? I think we did the right thing. So I think it was okay for them to stop. So, and of course, the New England Journal had this, thrombocytopenia and thrombosis, and with the AstraZeneca. So actually, the AstraZeneca is the one that kind of pushed this up. If AstraZeneca didn't have this and Janssen came on, nothing would have happened. But when two studies show, and they are both adenovirus, that gives you a little bit of a trepidation among the regulatory agencies, because God forbid, in another 5 years in United States, there'll be advertisements saying, did you take AstraZeneca vaccine? If you get that call, so and so and so and so and so and so, we can guarantee you that you will get paid, and we are not even going to charge, and it will not cost you anything. You see that all the time in the US. So that's what I think they were worried about, Okay, so so if you are concerned about the Janssen vaccine, it's this is with 6 patients that FDA presented. The median age was 33. It's about symptom about 8 days after the vaccine. So in fact, this week I had a patient who came and said, I'm having a headache, and she's a lawyer. So I was a little bit nervous, but this was about 4 months later. So I immediately brought her in, I examined, and I looked at her, and I said, listen, everything is fine. There's nothing to worry. This is way over this thing. And she has 2 kids. I said, your headache is because of your kids. Don't worry about it. Send them to a summer camp or something like that, or let your husband take care of the kids, and you'll be fine. And all those are white females. And then the platelet factor 4 heparin-induced antibodies were in about 5 out of 5. So we are actually tracking this, heparin factor 4, platelet factor 4 heparin-induced antibodies. We are tracking them to see whether there is any signal. But you can't go by this. Now, the Sputnik vaccine, I already mentioned that, they enrolled 21,000. The data is it's okay. But, what worried me was that if you look at the data at Lancet, please go ahead and, if you want, just go and look at it. The Lancet paper, if you Google it, it pops up. They censored some of the subjects from both arms, so for multiple reasons. So maybe there were valid reasons. So, but the reality is that it was reviewed by the Lancet reviewers, and they accepted it. 91.6% had efficacy, and the beauty is that it can be stored and distributed at -18, but you can keep for 2 to 8 degrees centigrade for global distribution. So I think Sputnik vaccine has a pretty good future. I think we need to keep politics aside and go for hard science and see what is right. And here's the data. The curves are the infection rates with the placebo is much higher, and you can see that just looking at the graphics, it looks pretty good. Again, I'm not going to compare. So here's the Oxford vaccine. Similar profile. I mean, between the 60 and 80s, you can't tell the difference. You get the picture that these things are pretty good. I mean, when I first started, I told you about the flu vaccine, which is like if you had 40% success, we were jumping up and down, and here we are getting 60 to 195%. So we should be delighted with the investment. And this is the Oxford vaccine. So again, the same data, looking at 74,000 people, 70% success rate, and it's interesting. Subgroup analysis. If you give half dose, followed by full dose a month later, 90% success rate. So this is one of the so this is very interesting because normally when you do clinical trials, we do dose escalation studies. So to find out what is the best dose, and we have time. We studied vaccines and drugs over 5 to 10 years. We rushed these 3 things through in 1 year. So as a result, we don't know actually what the doses we are trying, or the frequency we are testing, or the parameters we are looking for are actually the right ones. So I have a couple of slides later on to discuss this. So you have to be cautious about how you interpret data. But nevertheless, no matter what, all these things work very well. So don't get me wrong. I'm not saying that these vaccines don't work. No. They work like magic. But can you make it even better magic than what we have now? And these are the AstraZeneca safety. Again, as I told you that, it had a little bit of fever and fatigue. I know the British got a little bit finicky. I mean, if you came from like me in Sri Lanka, a bit of fatigue, just drink kotamali and walk away. But the British probably needed Tylenol. So again, the same thing. I'm not going to detail about that. So this is what you really have to concentrate. This is an excellent paper from Lancet, came in April. Efficacy, when you talk about a vaccine, is the degree to which the vaccine prevents the disease. The effectiveness, meanwhile, refers how well it performs in real world. So if you and I are not taking part in a vaccine study, then what you want is effectiveness. Don't tell me that it works in 95% on a study. I see this all the time in HIV. So they come up with a drug study and they pick these white males who are in the gym eating, healthy food, and they're doing all the right things and go to the gym and so mostly because the HIV more gay white males who are calculating their BMI and doing all that, and they get 95% success. You come and tell me that this works, and then I try this in an inner city African American low socioeconomic groups with mothers who are on drugs and mothers with 5 children and no social security or benefits. Nothing works. So efficacy is one thing in a clinical trial. Effectiveness is something else. So let's look at the data. So the ranking reported by efficacy gives the relative risk reduction, which relative risk means that how much risk was reduced by taking the vaccine compared to not taking the vaccine, which is the placebo. 95% for Pfizer-BioNTech, 90% for Gemalea, and 67 for Johnson, and 67 for AstraZeneca. So you can see that that this is this thing. So the relative risk reduction should be seen against the background of risk of being infected. And it varies from population to population. So I started off by saying you cannot compare two studies. So the relative risk reduction considers only the participants who could benefit from the vaccine in that cohort. The absolute risk reduction, which is a difference between attack rates and without vaccine, consider the whole population. So we propose the relative risk reduction of the study population should not be the gold standard. It's more absolute risk reduction. When you do that, when you take into the account the population studied, for example, Moderna and Pfizer did most of their studies in US. AstraZeneca and Gemalea and Janssen did a lot of almost 60% of the study paper came from Latin America and South Africa. So when you do absolute risk reduction, now the playing field gets leveled. It's 1.3% for AstraZeneca, 1.2 for Moderna, 1.2, 0.9, and 1.2 for J&J, and things get settled down. So the absolute risk reduction is also used to derive the estimate vaccine effectiveness, which is the number needed to vaccinate, NNV, to prevent one more case of COVID-19 as an absolute risk reduction. So if you go to the next one, if you look at the number of number needed to vaccinate to prevent one more case, you can see that to prevent one case, you need to vaccinate 76. To prevent one case with Moderna, you need 78 vaccines for AstraZeneca, 80 for Gemalea, 84 for Johnson. Pfizer, you need 117. So that's based on the modeling of the population risk. So when people say that, oh my God, BioNTech is like the gold standard, it's not really. When you look at the thing, these things all fall into place. So a number of number of people needed to vaccinate to prevent one case, you need to vaccinate more people with Pfizer-BioNTech compared to Moderna, or if for that matter, even AstraZeneca. So these are very interesting. So what these authors, Els Toratelli from Oxford, this came from Oxford, Nuffield from Oxford University, saying that if you don't look at the full picture, there's a reporting bias is introduced. So what I'm trying to say is that be careful of what people tell you, definitely in the internet. Assessing the suitability of vaccine must consider all the indicators, the safety, deployability, availability, and cost. And I'm not a public health expert. I don't think I'm an expert on anything, but basically that you have to look at these things and decide which is which basically comes to my next slide. This is the comparative effectiveness. So the relative risk is in red. So for example, if I take a Moderna here, there's a 90% relative risk reduction, but the number of people needed to vaccinate is 81. So the best, I think the best, based on this, just based on this, Moderna seems to be the best. And then, the other ones are like, for example, AstraZeneca, they don't look that bad. Pfizer, you need more people to be vaccinated to be effective. Also considering that they have to hold this cold. Although now Pfizer says that it can be kept in without freezing for a longer period. I think I should finish. So vaccine hesitancy. And you can see that there's great vaccine hesitancy among people in the United States. You Canadians are far more sophisticated than us, and the Sri Lankans and everybody else is more sophisticated than us. So the red are the susceptible, the blue are the immune. And basically what you want is, what you go at the bottom, 60 to 72% need to be vaccinated so that there is no transmission in dotted line, So even if they are immune, the red one susceptible, you won't get this one getting infected. So you need 60 to 72% vaccinated in a country to get to prevent spread. So these are the global data looking at when across the world, when people ask if a vaccine was available, would you take it? It's a little bit old, but you get the picture. United States did pretty badly. The best countries, if you take the so strongly agree and somewhat agree. So let's take the two blues. The two blues did pretty well in countries like Brazil, UK, Spain. These countries did well, and China also didn't do that well, because I think there's a great deal of suspicion of Chinese people about the government. But so you get the picture. So this is the tragedy about vaccine. You spend all the money and develop vaccines, and people don't trust those. And here is which best describe why you would not take the vaccine. So there are multiple things, like for example, US. Most of people talk about 30 adverse events and approval too fast. I think they are reasonable. Canada, again, very similar picture. Had the similar things. I think they are reasonable. There's no right answer in this, but it just gives you so the reason why you need to know is that when you pitch this to somebody, as a physician or a healthcare worker, when you're pitching it to somebody to say that, would you consider taking the vaccine, you have to understand why people didn't take, so that you have to address the needs, and then the question asks, what would when would you really take the vaccine? Is basically if the efficacy is over 19%, there's a high incidence. If the protection is more than 5 years, if major side effects are in 1 in 100,000, 1 in a million, and there are hardly any side effects, and if there's a complete use, not emergency use, then people will take the vaccine. So that's the kind of distribution. So now I'm going to go to the unanswered question that some of you colleagues ask. Only some vaccines have side effects and not others. Yeah, I guess, it just depends on the vaccine itself and the excipients, all these chemicals that you put in depends on how you're, and also it's your immune system, how some people have a very hypersensitive reaction, and, some people are just used to tolerating things. Should one jump at one chance to get any vaccine offered or wait for the vaccine his or choice? It depends. I think I personally would take the first one that comes, unless there are contraindications. For example, Janssen, the FDA is saying that if you're between, the women between, say, like young women, 18 to like the 40, try to avoid. It's a caution. It's caution, not saying not to. But I personally think that it's okay. It's okay. So I think that you should take the first vaccine that comes. So researchers have shown that vaccines are safe and better to have any vaccine available. Would there be will the mRNA change the genome? We don't think so. We don't think so. I mean, I know that I have the slide on the reverse transcription of the mRNA and getting into the DNA copy and getting into the host genome. But just because it gets into the genome, for example, if I get HIV, let's say if I get infected with HIV, so HIV viral replication, the HIV genome gets directly embedded in my DNA. So directly in my DNA, every cell which has a CD4 receptor gets infected and is in my DNA. Do I become suddenly does my nose start getting long like Pinocchio? No. I mean, do I develop dementia because I have HIV? No. I get dementia if my CD4 count goes down. I may get dementia if I get HIV perinatally before my organs are developing, and that could have effect. So just because a DNA copy of the RNA gets incorporated in the host genome doesn't mean that you're going to be some bionic man, or that the CIA can track you with geocoding from your RNA. That's really crazy. So, hello. So okay. So that won't happen. Duration of vaccine immunity. I already mentioned about the duration. Primary mRNA vaccines was symptomatic, and therefore I didn't. So these are the questions that that came from another group. Primary endpoint of vaccine was I can't remember that you guys asked. So primary endpoint of mRNA vaccine was symptomatic illness. Therefore, not. Yeah, that's true. You're absolutely right. These papers that gives the simple data should not be. The Lancet Oxford paper actually addresses that. They're saying that don't get carried away these hyperbolic promotional papers that try to tell you too much. So don't worry about it. Don't take it too seriously. Just take it for what it is. Long-term safety data requires years of vaccination. That's true. No data on children vaccine. Yeah, we are developing those. Less than 200 participant trial developed symptomatic COVID. 250 drug control. Absolutely right. We need larger studies. But I don't think anybody is going to do a study. What we need to do is like a phase 4. So we need to do cohort analysis over time and then see with the real world data. So the last point is excellent point. Somebody asked, the genome appears relatively stable, but not know when you add selective pressure what happens. That is very true. So this is why I worry. So when we were giving convalescent plasma, we were slightly worried whether if you put selective pressure, whether the virus will move in a different direction. That is very normal for a virus. So if you're in HIV, that's why we give 3 drugs in HIV, because if you give 1 or 2 drugs, the virus can mutate against those. But because virus always trying to escape. So that's a very good point. So we have to be careful. So again, a thing about reverse transcription, I'll talk about it later. You can mix different virus. It doesn't really matter. If you could address the topic of mixing different brands of vaccination, because the final common pathway is the mRNA. So whether you use adenovirus or mRNA, common pathways are mRNA. So it won't matter. Out of several vaccines approved, what are the types of appropriate tropical climates? I think the Janssen looks one of the best. Janssen, maybe Gemalaya, maybe the AstraZeneca, which will be okay. So the next one is okay. So this one I talk about reverse transcription. You what they did was that they showed that that COVID-19 RNA can be reverse transcribed and integrated into the genome of the infected cell. And they looked at some other chimeric transcripts on the patient-derived tissues. So not only that they managed to put this in, but they looked at postmortem tissues and other tissue, and you can see that some of these are integrated into the host genome. So it doesn't, as I said, that doesn't mean that it's a big deal. It's not a big deal. Okay. So now let's quickly the variants. Variants are spreading everywhere. I told you why the variants are happening. They mutate. They are RNA viruses. They're supposed to mutate. So sometimes, there is a survival advantage. So there's a faster transmission. B117 is from United States. Sorry, United Kingdom. B351 is 1351 is from South Africa. So, and then there is a preliminary evidence that is increased severity, right? And sometimes you can avoid diagnostic tests because there's a thing called S-drop. In the PCR, certain segments of the PCR may not work. But generally, all the PCRs have taken this into account. So that shouldn't be a problem. So decreased susceptibility to therapeutic agents like monoclonal antibodies or convalescent plasma, that can happen. So evasion natural vaccine immunity, that can happen too. So, for example, B1351 has 10 to 12-fold low victory vaccine efficacy. So again, the Brazil variant is called P1. They have a similar issue. And you can see that, when you have a hypothetical case of this type of variants, more transmissible variants, death from strain is increases as you go on. So, okay, so I'm not going to already mention about that. So one thing is that these variants are going off everywhere. It's not coming from Brazil. It's actually spontaneous mutations happening in each area. Instead of Florida, we have every variant you can possibly think of. So this is, again, this slide came from Tony Fauci's talk a couple of days ago, showed that, the B117 is covered by J&J, 1351 from South Africa covered by J&J, and then the Brazilian one, we are not sure. The rest of the things we are not sure which vaccines work, but at least you know that the UK and the South African variants are well covered by most of the vaccines. So finally, almost my last slides is to date, the reported safety and efficacy have exceeded expectations. But I agree with most of your questions that we have to be cautious. But perfect is the enemy of the good. So don't look for perfection because then you will miss the good. You have to talk about the effectiveness rather than the efficacy because at the end of the day, we live in the real world. And of course, we have to make sure that these vaccines are distributed equitably to all the countries like Sri Lanka, Bangladesh, Pakistan, India, and and Africa. And vaccine hesitancy is a problem, not in Sri Lanka, thank God, but other countries. And of course, the variants may affect the efficacy. Finally, there's a race, and COVID-19 versus vaccines. I think we are going to win this. This slide actually was shown by Tony Fauci a couple of days ago, and I stole it from him. So with that, I'm going to stop and take any questions, and thank you.
Thank you, Dr. Javir. That super presentation. And I think there are lots of myths around this whole subject. And the beauty of your presentation is that you presented a lot of scientific truth, scientific facts to, I think, go against these myths. I think that is the expectation of your presentation. I think you did it marvelously and super. Thank you so much. And also, I think you have taken a lot of the questions that the participants gave us before today. And there are a few, I think, questions that are there which came after we sent you the questions. I think some of these questions also answered. Now, there's one question remaining, which I will take, and then I think Harini can take another question one or two after this one, which are the questions that came after we sent you the questions on, I think, Monday or Tuesday. I'll take the one that has not been addressed. Now, this one is, are the vaccines being rolled out effective against all the variants of COVID-19?
So at this moment, it's very hard to answer because the variants keep changing every day, because the virus keeps mutating. So the general answer is that most of them work. So that's why that I am a bit nervous about the CDC saying take off your mask. I tend to keep my mask on, even if we are vaccinated, because of that possibility. So at this moment, we could say that with what we know, it works. But what I say could be wrong in another 1 week.
I see. Okay. All right. Then, Harini, would you like to take the next question?
Yes,
The question to you, yeah.
yes. So the next question is, I think from the audience is, because Sri Lanka is forecasting like increased number of COVID patients, they want to know what's the best strategy to reduce the incidence of the cases.
I think we have to go down to basic principles. It's that we have to wear masks, do the simple things, and shut down. I mean, it's very hard to shut down cities because then the country has to go on. The country is in so much debt. It's very difficult. But at least you should minimize. Whoever can work from home should work from home. All the companies, we have to decrease the traffic encounters and all that. That's the first thing. Then second thing is to vaccinate. So that's why that we all, whatever the resources all of us have, uh, we have to try to support the Sri Lankan government to access vaccines and distribute it. And and also we have to make sure that the Sri Lankan government, thank God they are working with the College of Physicians right now, because most of my communication with Sri Lanka is coming from the College of Physicians. They are telling the government what to do, which is actually a very good thing, so that we can uh, make sure that the vaccines get delivered, and we have to make sure that they have adequate PPE and all that. Beyond that, there is very little you can do. I mean, we can treat the cases, we can isolate the cases, we can do all that, but these are the simple things we can do.
Thank you, Dr. Javida. So next question, I think Anura is
Yeah,
going to take.
that's right. And actually, there is a question to our, the, not the chat line, this has come to WhatsApp group. Now, this is about Pfizer's second shot. Given at 4 months mark, is there advantage in the immune response if you keep the 4 months gap between the first and the second?
So that is very difficult to answer because there are no studies. There are no studies to answer the question, because whenever you say it is good or bad, we have to have a control group. So and then the control. I mean, so I'm going by first principles of clinical trials. Clinical trials has to be first designed that way. But if you, I mean, let's say that you know, if you find that more people are doing well, 4 months looks little too long for me. But the problem is the logistics. We have to be realistic with what we have. So I think some of the governments are pushing to give the first vaccine first and get as many as possible covered, and then they give the second vaccine later because of the logistical reasons. So I guess, without second guessing, that may be a strategy which may work, rather than the opposite, which is like vaccinate less people, but people who you vaccinate have a good, robust system. The only thing that tells me that 4 months may be okay, because after one vaccine, there's almost like 80% uh, con- uh, kind of protection for fairly long time. So I think the people, I mean, I know why the question was asked. I think there is a logic behind that decision.
Okay. Thank you, Dr. Deshante. And Harini, you want to come in?
Yeah, I think there are lots of questions, and Dr. Javira, I think you stimulated the audience. So one is about, I think the mix and match of vaccinations. I think this is relevant to Sri Lanka and also to Canada because people in Canada got the AstraZeneca vaccination and it was a temporary pause. And in Sri Lanka, I think after giving the AstraZeneca, they ran out of the vaccination. So if you can briefly talk about the mix and match vaccines. Thank you very much.
I think you broke up a little bit, maybe my internet. So I think mixing and matching is okay because I mean there are no studies. Again, you have to be very careful. I mean there are no studies, but going on first principles that if you can't get the same kind, get something different. I mean this has been asked from me by a lot of my Sri Lankan colleagues, and I said go ahead and take the. I mean if you want AstraZeneca, go and take Sputnik. You know it's okay because they are both adenovirus. I mean, it's not going to make a huge difference. So now in Canada, different. Like if Pfizer was delayed by 4 months, that's a different strategy. It's hard to say which is better because there are no studies. Because whatever I say could be wrong later on. So that's a personal choice you make. Either you take a different vaccine at the right time or wait for longer time and get the right virus, right vaccine. It's a very difficult question to answer.
Okay, I suppose we are in the process of, you all are in the process of collecting information
Yes,
to respond to these questions
yeah.
in the future. Thank you so much for that one. And there's this interesting question. I think it's like a question was put as a personalized question. I'll read the question and I'm sure you should be able to grasp it. It says, I have volunteered in the past to a vaccination trial that used chimpanzee adenovector. Will that make AstraZeneca vaccine less effective on me?
Yes. Yes.
I see.
Yes. I mean, I think because if it's the same strain, it could be less because you already develop antibodies against the chimpanzee adenovirus. So now this I'm going by basic first principles. Basic first principles. Like if you've been vaccinated, the reason is that even if your antibodies are low at this point, because antibody titers wane off, but what happens is that there are cellular memory cells. So as soon as the chimpanzee virus comes back, the memory cells, oh wait a minute, there's a chimpanzee virus here, and it starts producing antibodies. So it could happen. It could happen. So if it was me, I would consider going with Pfizer or Moderna. Or even Janssen, because only AstraZeneca uses chimpanzee.
Oh, I see. Okay.
I mean, this is basic on first principle, I mean, it's not a health advice, but you know what I mean.
Thank you. Thanks. Harini?
So, Dr. Javira, the next question is, I heard that the infected people with no symptoms are really big. How big is this? It is really big. How do you take such people who have some immunity in the calculation of the herd immunity?
That's a very good question. So it is calculated in, first of all, it's very hard to quantify this unless you do the population surveys. So that's a very good question because if you assume that 20% of the population was already infected, technically they have some immunity, so therefore that should contribute towards herd immunity, depending on the robustness of the antibody response. Even if you had a mild infection, so when you have mild infection, your immune response is not very good. Your T cells, memory cells, are still there. So actually, that's a very good question. I mean, you could consider, you can add, potentially, you can add that to the herd immunity equation. So I'm not a population scientist. I'm sure they are considering those.
Thank you. Thanks. And there's another one in the chat line. I'm not sure whether I think you touched this a little bit, but I will read the question. Are there any long-term side effects expected from mRNA vaccines or vector vaccines for COVID? Long-term side effects.
Yeah, it is, it is hard to say because so you have to go by the history, right? People think that mRNA vaccine came last year. No, Moderna has been working for about 15 years, and Pfizer have been working on this for a long time. So they have been putzing around with this for a long time, and then adenovirus vectors have been for 4 decades. They have been working for 40 years. So we haven't seen some really nasty side effects from this. I mean, clearly, some people have done Guillain-Barré, some people have neurological problems, like any vaccine, but there is, the people who are against vaccines have said that, you can have neurological problems, children can have ADHD and all kinds of things. I mean, it's very unlikely. So at this moment, there is no, unless what is classically reported by vaccines, I doubt whether these things would cause long-term side effects. But we don't know. We don't know because, again, it's hard to say. But I think they are pretty safe at this moment.
Thank you. Thanks a lot. Yeah.
So, Dr. Javira, the next question, and thank you for answering all these questions briefly and in meaningful way. So the next question is it ethical to use placebo for this kind of research?
Very good question. Very good question. Okay, so first of all, you need a placebo group, right? You need a placebo group. Otherwise, you don't know. I mean, like this is what happened in Sri Lanka. Like, they were having some Ayurveda stuff and they were trying to run through without placebo. You can't do that. The second thing is that AstraZeneca did a terrible mistake by having one study they did without a placebo. They just had a control group randomly selected. There was a study with convalescent plasma with UK did through the National Health Service, didn't have a placebo group. So you always get the wrong results. So you have to have a placebo group. But the question is now, the next study, should it be against the placebo or should it against the comparator? So from now on, you can never ever do a COVID study with a placebo. It has to be compare with the best, which is either Moderna or compare with the standard of care. So so FDA doesn't tell you what is best. FDA says we have approved once Moderna, Pfizer, and Janssen gets approved, then that becomes standard of care. So next vaccine that comes to get approval have to show they are either equal or better than the standard of care. No more placebos. No more. So that's important. About the vaccines the design. In fact, there was another design called challenge studies, which British were doing. That means that you take the lowest risk people, say like 1,000 people, and you actively infect them with the corona. You vaccinate and infect them. I think, Andrew, you were there at one of my talks. I talked about that. And then actually it's pretty ethical. It was approved by the MRC, and because it can save lives and you can get a result very quickly within like 3 months. So those are different designs.
Good. Okay. Thanks. So there is another, I think, a technical question. Of course, all these are technical questions, but this is coming from, I think, a researcher. What may be the reason for actual infection is less immunogenic? I don't know whether I pronounced it correctly.
Yeah, that's correct.
Than the vaccine.
Yeah. So, well, it's hard to say because sometimes the vaccines have a kind of adjuvants that will boost the thing, boost the infection. So it all depends on the individual. So the question is actually very good because I cannot stand here and say the vaccine gives a better response. No, That's not true, because some people will have a much better response with with the uh, the infection than with the vaccine. Uh, so I cannot say. So I would never say that vaccine is better than the real infection. My comment was that if you had the infection and if you had the choice of getting a vaccine, I would take it rather than saying, well, I already had the infection, so I should have enough antibodies. So I would never say that the one is better than the vaccine is better. I mean, the question is actually very good. There is nothing to say that this is better.
Thank you, Dr. Javed. Yeah, Harini.
Yeah, so the next one is, what might be the explanation of the gradual age shift of the group with higher infection rate? Example, high rate initially in the older people versus currently in the younger population.
Great question. Well, there's this saying in Sri Lanka that the water flows from the lowest side. වතුර බහින්නේ අර පහළට වගේනේ. You think so? It goes when the weakest people gets first. So as you get older, although we think we are the strongest, we are the weakest, especially men, right? Okay. So as you get older, your innate immunity goes down. So when an infection comes, it hits the older people first. So now you have kind of that up, and most of them got vaccinated. So now you have developed a kind of a sort of a herd immunity among the older people. Now the younger people are getting the infection because the virus spreads from susceptible person to susceptible person. Now, and also the variants have come, and so the infectivity has gone up. So they are more aggressive, infectious, and of course that's the population that is available. So that's why that it is like that.
Okay, thank you. And you have actually created a lot of interest among our participants, so there are questions coming up. The next one is, are there any clinical trials data currently available for COVID vaccines for children, pediatric trials?
Yeah. Sorry, say it again.
Sorry. Are there any clinical trial data currently available for COVID vaccines for children? That is pediatric trials.
No, not at the moment. No. I mean, I'm sure they are developing, but it's to my knowledge at this moment they are not. But I think I'm sure they will come soon.
Okay.
Yeah,
Thank you.
Anura, I have a call from the hospital. I'll have to.
Okay, sure. All right. So let's call it a day at that point with your presentation and question and answer session. So thank you so much, Dr. Javida. So you need to move out, I suppose.
Yeah. You guys carry on. Thank you so much. You've been a great audience. I love the questions.
Yeah,
These are amazing.
Thank you ever so much. And yes, we will have a couple of items to sort of to deal with the audience. So we will do that. And yes, so please get on with your busy schedule.
Okay.
And
Okay.
this is
Thank you.
very fortunate. Thank
Take
you so
care.
much.
Thank you.
Thanks. Okay, and yeah, we have great attendance so far. So before ending, before going into the vote of thanks, we have two messages for the audience about our future plans and future activities. First one is this the important message from Dr. Javeda, who just presented this webinar. And I think during the speech, he just mentioned about the work that he's helping in Sri Lanka. That is currently he's leading a fundraising effort to collect some funds and send to Sri Lanka. Particularly, he's aiming at I think this Gampaha or some hospitals. And he had got a list of items for to be helped with funds. So our message to the audience is that we will send you an email with all the details. and also the way that if anybody likes to contribute to the funding, the way that you can way to transfer funds for this purpose. So we will send that email with details, and once we get the let's say how to transfer the money and all that, we will inform you. But any of you wants to contribute to that effort, the timely effort and the good noble cause, so that you have the option to do so. So please wait for that email, and we will be sharing it very soon. That's the first message. And the second one is this walkathon. But I think many of you know that we used to have this health walk annually. Actually, this health walk was initially proposed by Dr. Harini Silva, who moderated our session today. And but now we cannot do this because of the pandemic, and therefore, as a health activity, we are proposing this walkathon as a challenging event. And this will most probably happen in the month of June. And there are also, now you can see the flyer that we have developed, giving all the information and the details and how to register and what's to be expected from you and so on. And we will be sending an email soon again with all the details and to register, and giving also the information as to how you can register. And mind you, this is a competition, and if you win this by having, I think, the highest number of steps by walking, you will get good prizes. So let's stay tuned and wait for the emails with all the details of this event. Very soon we will be sending. So those are the two messages I want to share before we conclude this webinar with our vote of thanks for the webinars. And first, I think on behalf of everybody, even though Dr. Dushant Javeri is not here, we want to thank again for his valuable time that he spent with us, and also his valuable information. I think that has, I think, taken out a lot of the myths and all that we were having in our minds. So thank you so much, Dr. Javeri, for your time. And then let me thank our coordinator, Ajit, who did a good job of coordinating everything from the day one to get the webinar going. And he's behind the screen and did a lot of work to get this up and running. So thank you very much for your hard work, Ajit. And we had a lot of artworks for the flyer to give the popularity, and as well as the countdown work. And there are a lot of efforts in this. So Deepani was very generous in putting her time into this and organizing everything. So we are thanking her for her time. And Niroshan actually showed his very innovative skills by putting things together, particularly in the countdown. And thank you so much, Niroshan, for that. Then I would like to thank the members of our executive committee. In different times, we had to interact with them to get various ideas and polish our ideas. So they were very helpful and very prompt in giving their messages. So thank you very much for that. And Manoj, you are the IT director of the whole affair. So you did a lot of things to put these things together, and also to run it without any hiccups. So thank you so much for your efforts, Manoj. And finally, of course, not making any belittle our the participants from all over the world. And because of you, we are actually enjoying this, as well as we are interacting with you very effectively. So I thank you so much for all the participants and the community Ottawa and everybody from other countries for your was moderating this session with her busy schedule. And thank you so much, Harini, for coming forward. And finally, let me very thankfully recognize our two sponsors for this webinar, Murugesh Gandhi and also Vedantika D. Lambert. And thank you so much, both of you, for your generous financial support for our work. And as you know, this all this money goes to the needy courses that we are sending these scholarships to Peradeniya students. So we are using this the funds for that. Now we have come to the conclusion of this webinar. I think it's a great pleasure having all of you with us, and we probably hope to see you very soon again with various activities that we would be organizing. So stay tuned for our maybe next webinar and next activities. So thank you so much.